Get started here in a second. Let me test the audio, as I do compulsively now that I forgot the audio one time. All right, audio is working. I can hear. I will usually play the stream on separate YouTube account just to make sure that externally all the audio works and the lag is fine and so on. Alrighty, today we got some espresso coffee here. And we're going to close out this week, this ChatGPT week, and we're going to give ChatGPT its hardest challenge. So, yesterday we were doing a Kaggle competition using only ChatGPT, and it actually worked quite well. We were able to get to a final submission, and it didn't break any records, but the fact that we were able to get there was pretty impressive. So today we're going to try to do kind of a step up in competition. So the previous competition was Spaceship Titanic, this one here. I mean, this is a tabular binary classification problem. So, I mean, fairly simple. Basically, the solution that we got was just a bunch of pandas code. And now we're going to be trying a step up in difficulty, which is, this is Enzyme Stability Prediction by Novo Enzymes. I don't know what Novo Enzymes is. Maybe it's like some kind of Novozymes. Probably a startup. Novozymes is a biotechnology company from Denmark. Founded in 2000, so 20-year-old company. 15 billion in revenue. They have a challenge here for predicting the ranking of protein thermostability. So this is now more difficult, right? Because it's not just a matter of plugging all of these into a pandas data frame and then training an sklearn multi-layer perceptron on it. Now you're dealing with proteins, right? There's some kind of, you need knowledge about biology and kind of the proper way to deal with this input data. And ChatGPT, one of the things that it does is it knows a bunch of things. So maybe we can even ask it some generic let's ask it something kind of generic about the data here and see what it has to say. How is the stability of a protein? Let's ask it that. How is the stability of a protein to temperature changes determined? There are several ways to determine the stability of a protein to temperature changes. One technique is called differentiable scanning calorimetry. Measures the heat absorbed or released by a protein as it's heated or cooled. Okay, another method is to use spectroscopy, such as circular dichroism or fluorescence, to measure the changes in the secondary or tertiary structure of the protein as it is heated or cooled. Additionally, the stability of a protein can also be determined by performing a thermal shift assay, where the protein is incubated at a range of temperatures and the amount of active protein is measured at each temperature. Okay, so ask it. So are you telling me that a protein's response to temperature is non-linear? The response of a protein to temperature changes can be non-linear, meaning that the protein may not change in the same way or at the same rate as the temperature changes. Quite smart. Protein may remain stable over a certain range of temperatures, but then undergo a sudden change in conformation or function at a specific temperature. This change in the protein's behavior with temperature is known as thermal transition and can be measured using techniques such as DSC or spectroscopy. All right, pretty cool. So it does have some biology knowledge, so good to know. Hopefully it can maybe use that to help us. So maybe before we even start looking at this, let's go ahead and get our developer environment set up. So we're inside the Kaggle folder of our Betterer repo, and you can always go onto my GitHub po, and I have this repo Betterer here. It is a public repo, so you can go ahead and find this code if you want to. Specifically, there is a Kaggle folder in there, and we are going to make a new folder, and we're going to call it, let's call it enzymes. We'll go to enzymes, and I think we env environment or env list. env virtual ems. So I think we have ESM protein over here. Yeah. So we did some streams a couple weeks ago for protein folding and protein prediction. We used what's called ESM2 to Meta AI. ESM2 this. Yeah. So evolutionary scale modeling 2. This is basically, you could think of it like the ImageNet or the CLIP for proteins. So basically they have these huge databases of proteins, and they're folding and kind of how the different chemical sequences, how they result in the shape of the protein. And they're trying to usually predict these problems of like predicting the shape given the amino acid sequence. So for example, a protein might be might look like this, right? It might look like this string here, where it's just a string of letters, and each of these letters represents an amino acid, and then a protein is basically this sequence of amino acids. And the way that these amino acids cause the protein to kind of fold onto itself is kind of tricky to figure out, and there's a lot of different factors that go into it. So basically a lot of work is done in figuring out how to make that nice. So basically they train these large models on huge databases that are usually government databases, such as like the National Health Service in Britain has a huge one. And then they have these giant pre-trained models. So what you want to do, or what we're going to want to do for this problem, is we're probably going to want to use this, for example, ESM2 15 billion, this large model here, to take the sequence and then turn it into a feature vector that kind of has some semantic representation of that has a latent representation of that protein in embedding space that kind of has that has semantic meaning. And I think that ESM protein atlas, this is the latent space that we want right here. So this is what the like latent space of this ESM model looks like, right? And you have all the different proteins here, and they all map to this latent space, and you can look at the distance between these, and things that are closer are going to be related, and so on. And I assume, and we don't know for sure, that things that are close together in this latent space probably have a similar temperature response because their structure is similar, the way that they fold is similar. So I think that's probably going to be the best way to go forward here. And they also caveat here. They say there are publicly available methods which can predict protein stability, such as ESM, EVE, and Rosetta, without using. These methods may also be used as part of the competition. So you're allowed to use ESM, and I'm pretty sure ESM is you can use it. It's MIT license. Yeah, MIT license. So we're definitely going to be using this. All right. So going back to our developer environment setup here, we're going to be using the ESM protein environment that we created before. So let's go ahead and IAM activate ESM protein. All right. Now let's actually look at what they include. So let's hit join competition first of all. Understand and accept. So there's a couple different files here. You have train CSV, which is, let's download it while we're talking. Train CSV, download. Test CSV, download. Wait up. I think our download folder has a bunch of crap in it. Let's go far. Remove -rf star inside that folder. Now let's download it again. Download. 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 Okay, and then there's train updates, corrected rows in train. Okay, so there's some amount of corruption in the data, but we don't necessarily care about that. And then wild type structure prediction. Okay, I think just with test and train we should be. And sample submit. Okay. So we're going to go ahead and copy these into here. We're going to copy downloads test.csv. We're going to put it in there. We're going to copy sample submission and here, and then we're going to unzip downloads train csv.zip, and we're going to dot let's see how big that is. 15 megabytes. Okay. So now let's go ahead and exit out of that terminal, and let's open up VS Code in this folder here. All right, so if we look at train CSV, have our training data. Marketplace has extensions. Is Rainbow CSV. Okay. Now we have Rainbow CSV. Seems like VS Code extension. I think it changes the color so it's easy. Yeah, there you go. So nice little extension here, right? You have the columns here. You're going to have sequence ID, protein sequence, pH, data source, and then TM. So protein sequence is the amino acid sequence of each protein variant. pH is the scale used to specify the acidity of an aqueous solution under which the stability of the protein was measured. Okay, so if you remember, ChatGPT here was saying that basically the response of the protein to temperature is nonlinear. So basically they're putting these proteins probably inside like a little liquid and then heating the liquid over time and then measuring how the amount of protein changes or something like that. So this is probably just the pH of the liquid that was used when they were measuring that. And Be curious if always 7.0. Yeah, it looks like it's always 7.0. Okay, maybe not. You have here some that are 5.0. So some people are measuring the protein stability in like a slightly more acidic. Data source. The data source where the data was published. This is interesting. This kind of tells you that there's going to be different maybe organizations, different data sets that these are from, and then that's going to that is yelling out. Whoa. I'm going to see it might be stuck. I'll be back in a second. It was stuck. My cat will go into the bathroom and then push on the door and lock itself in the bathroom. So not the smartest cat. Okay. Sample submission and test. So there really isn't much here. There isn't a lot of different features. Kind of just have the sequence itself. And that's large hit. Okay, let's look at the sample submission. The sample submission has sequence ID and then TM, which is I think the target column. Where TM is not an actual temperature, I don't think. I think it's just a temperature score or something like that. Higher means the protein variant is more stable. If TM goes from 0 all the way to 24,000 or 2,400. Okay, so this is a regression problem. You're trying to go from a bunch of different inputs, mostly just the protein sequence and the pH and the data source, and then the final output of your neural net is going to be a single number, which is the target, or which is the temperature max. They actually say thermostability. Oh no, okay, melting point. So these melting points are a little bit more, they make a little bit more sense. 48, 43, 65. Okay. So let's go into our terminal here and let's just install some basic ones. Back to stay in our. Let's make a notebook here. Grep our history. Pip install lab. So now we have our Jupyter lab in there. Let's open up our notebook. Let's put a little markdown cell above this. Hash. Plus mark. Say stream. Enzyme Stability Prediction. Kaggle with ChatGPT. Link to this stability prediction here. So link ChatGPT. Now let's ask ChatGPT here. Can you write Python code for me that reads two CSV files, train.csv and test.csv, into a pandas data frame. The CSV files already have column headers. Let ChatGPT work. All right. Go, it's reading the CSV files, putting them in the data frame. And it says the read CSV function automatically assumes the first row of the CSV, so you don't need to specify this. Run that. Got it. Can you print or can you write Python code to print the first couple lines of each of the data frames? I also want to know what the data types of each column are. That. Okay, it's actually repeating previous. Here you go. Now it's doing the head command, and then it's printing out the D types. Copy paste. Look at this little kitty. Go ahead and print these. Make this a little bit. Okay. So we have our columns here. We have the sequence ID, protein sequence, the pH of the data source, temperature maximum. That's the train. Then we have the test is basically the same. And we have the data types here. We have sequence ID as an int, protein object, pH is a float, data source is an object, EM is a float as well, and the test data has protein sequence, pH, and data source. Okay, so I guess maybe the first thing we want to do is can you write Python code for me that checks what percentage of the rows in this pandas data frame have null or nan values? Okay, here we go. So here we're going to have to replace this with f train df is what and we'll call this. And then if we Copilot auto completes and does the have print train null percentage, and then we have the test null percentage. So a lot just happened there. If you aren't paying close attention, I copy pasted code from ChatGPT, and then I used a lot of auto completion from Copilot. So I have GitHub Copilot on this VS Code instance here. So 2 different code generation, code completion, whatever you want to call it, AIs playing with each other, and I'm kind of interfacing between them here. So quite crazy to think about. So let's go ahead and run that. Okay, so you have here for the train, you basically have no nulls in the sequence ID or in the protein structure. You have a very small amount of nulls in the pH, a very small amount of nulls in the data source, and no nulls in the temperature maximum. And then in the test data, you have no nulls or nans. So go ahead and remove these. Can you write Python code to remove any rows from 2 pandas, or from a pandas data frame called traindf that contain nan or null values? Sure, here's some sample code that should do what you're asking. I wonder why it keeps like giving us this first part, you know? Okay, I don't want to do this in place. Can you make the result save it, or can you put the resulting data frame into a new data frame called traindf filtered? See if it gets that. It's interesting. It says, let me know if you have any questions about this. All right, perfect. Put that in there. So now we have our filtered train DF. Okay. So next thing we need to do is kind of want to plot the pH value here. So want to see what the pH. A little bit tricky. Can you write Python code that plots a histogram of the of all the values for the pH column in the of all the possible values for the pH column in the train DF filtered data frame. So we're doing a little EDA, exploratory data analysis here. Sip of coffee while it chugs on that. Okay, it's using matplotlib. We might not actually have matplotlib. Let me do it. Matplotlib. And then you have alrighty, so you can see here that can you add a title and add a title pH to this plot. I also want a label on the x-axis that also says pH. See, there's an interesting thing here. It kept repeating the code, so it keeps repeating this reading from the CSV file and the import pandas. But then once we asked it to plot, it also still repeats the CSV file, but it doesn't repeat the pandas import anymore. So this code is actually wrong, right? If you tried to run this whole code, you wouldn't have pandas as a dependency. Or you would need the import pandas. So it's interesting how it's kind of losing track of some of the earlier context in the conversation. Okay, so here are the title and x-axis label. There you go. Got the pH now. Let's see if we ask it, can you modify the above code to plot the PM column instead of pH? Let's see. Okay, so I asked it to plot the temperature maximum column, but I'm curious as to whether it'll also change the title and the x-axis label. Because I didn't explicitly ask it to do that, but logically you would do that. So see what happens. Okay, so it's not doing the pandas import anymore. It's still doing the matplotlib import. It's still kind of doing this boilerplate drop NA that we had it do earlier. It is correctly changing the column to TM. Here's the, yep, there you go. See, it's actually quite clever right there. So it replaced the labels on the title in the x-axis. Even though I didn't explicitly ask it to do that, it understood that that makes sense. All right, so we can go ahead, copy, paste that, put that in there. There you go, that's the temperature maximum. Okay. So what else do we have here? We have the sequence, the pH, the data source, and the TM. The data source, how many possible values are there? Can you write Python code that finds all the unique values for the data source column in the train df data frame? Let's see what it comes up with, because I think the problem is that the data source is going to right, some of them are just going to say novozymes, but I think some of the other ones, if we actually look at the screen, the data source is more complicated, like you have DOI. We'll see how many are here. Oh, look at that. Now it's doing the import pandas again. What if we actually tell it that you don't need to keep importing pandas and reading from the CSV? I already have that written in my code. From now on, just provide the lines of code that I ask at the so let's print that out. Let's see. Okay. So this is we actually size of that. Print the length of the result. There's 325 possible difference. Look at that. I apologize if my previous answers were not helpful. And then it goes ahead and it gives me exactly what I wanted, which is just this part here without the import. Perfect. Okay, so now that we've done a little bit of exploratory data analysis, we're going to try to do the hardest part here, which is, this is going to be very crazy if it gets it, but can you write Python code for me that uses the repo and loads one of the pre-trained models on there. Models that they have on this repo. I specifically want the First of all, let's see if it just gets that. We're giving it a GitHub repo, and we're asking it to load one of the pre-trained models in that repo. So ChatGPT does not have the ability to like actually go out and like get information on the internet. Yeah. Okay, so there you go. I'm not able to browse the internet to access the repository. Yeah. So, and you write Python code that uses the let's actually give it part of it here from the examples, and then we'll ask it to complete it. So can you write or complete this code for me? I want to use this model to convert my protein sequence strings into feature vectors. Try again. Might have just killed it. Edit. I think we might have just, oh no. Oh no. An error occurred. All right, guys, I think we might have killed it. Unfortunately, it seems like maybe here when I asked it to go on this website and look at that, we might have gotten flagged. And now it just might be blocking us out because it thinks we're doing malicious things. So let's see if maybe we can use VS Code instead. So let's import ESM. No module name ESM. No good. Pip install fair ESM. ESM fold. What? Does seem like we have ESM, so why doesn't it? Oh, because we're not using the ESM protein. All right, there we go. The clear output of all cells and save. Import ESM here. I think we also need torch. We have both torch and ESM here. Good. And load the ESM2 model. Let's see what VS Code tells you. Okay, so VS Code wants to use the 650 mil UR50. Okay. Model dot eval. And here they have a bunch of batch token representations, batch token, return stats. Okay, so I think right, what we're probably going to end up doing is we're going to create a new column in the pandas data frame that has all of the tokens from this. So let's go ahead and we'll do the batch anyways. So maybe not, you know, like I don't we necessarily need that. I think we can make a function here. Define convert token. Or let's call it encode sequence. Code the sequence using the ESM2 model. So actually, here you go. Batch converter, the sequence to CUDA. With torch no grad results. This might actually even be the right thing. Like that is quite crazy. So, damn, look at VS Code here. Or look at Copilot. Encode the first 1,000 rows of the sequence column in the train data frame. Train encoded equals train df sequence head 1000 apply sequence. Let's see if we say into a new column. Okay. That is, hey, maybe we didn't even need to be using ChatGPT anyways. I think actually secretly these are both the same model, like low key ChatGPT and GitHub Copilot. They're, I think, right, OpenAI trained both of these. OpenAI trained all of it on Microsoft Azure. Microsoft owns GitHub. So these models might be much more similar than different, right? They're kind of made by the same people. They might be different versions. The version that's here on VS Code might be a smaller version. The version that's on ChatGPT is maybe fine-tuned to give you not specifically coding stuff. So they might not be the same exact model, but they're very, very similar. So they're kind of related. Okay, so let's go ahead and run. ESM pre-trained has no attribute, blah, blah, blah. I think it's going to want this one. Okay, so it's downloading the model, putting it in the torch cache torch hub checkpoints, and we can actually go and check out how big that is. So we can copy this code here, open up a new terminal. This here, ls ht. Okay, so you have this model is 29 megabytes. This ESM T6 UR50. And we can actually go, they probably have an explanation of what these are. So ESM2, T6, 8M, UR50. Back to view. What is it saying? Name, train DF is not defined. Oh, okay. Outputs, that, this, run this, that. Now let's key error sequence. What is it actually called? It is called protein underscore sequence. And we're going to call it ESM. We might have to convert protein sequence into a call. Got an unexpected, and that too many values to unpack. Expected to. Okay, so the raw batch. We're giving it the sequence. Right, so here in the example here in the ESM repo, they the data that they feed into the batch converter is a list of tuples, but here we're giving it the sequence. So maybe we need to do this. We need to give it a dummy variable here, like that. So we're giving it a dummy tuple with a dummy index. Because I think what it did is it took the sequence and it just unrolled that string. That's why it says too many values. Let's see if that works. ESM object after star star must be mapping, not to. Okay, so the batch converter takes the data and then outputs three things here: batch labels, batch strings, and batch tokens. Let's actually go ahead and explicitly do that. And then with Torch Nograd, you just want to give it the batch token. And results, representations, key error 0. So there's no 0th element of representation. Maybe you don't need this 0th, maybe this. So, Manuel Oksana, hello. Dict object has no attribute squeeze. All right, so let's import PDB and then pdb.setTrace. We're going to run that. Okay, we're inside the PDB. Let's say results. Okay, so results representations is just an empty dictionary. So if we say results logits, and then let's ask, right, then you have a tensor. So results logits, and then say dot shape, so we can get the shape of that. Results, logits, results, logits, or logits. Okay, so it's a 1 by 343 by 33. Yeah, so if we actually, residue is the amino acid residue, which is basically a single one of these. So we want a vector that represents the whole protein, right? So we want to do this step here, where we're actually not just getting the token representation, but we're also getting the full sequence representation. I wonder why representation. What is this repr layers? 33 return context true. I wonder what happens if we add that in there. Interrupt. Save. Run this again. Gonna PDB in us again. I'm gonna print results. Results next. Okay, so now results has the answer. Do results representations 33. Okay, you get this tensor. Results represent 33 dot. It's a 343 by 1280 matrix. Okay, so let's go ahead and do copy this. That token representation. Then we're going to do the per sequence representation. What is batch lens? Batch lengths. And you get that. Put another input. Okay. Interrupt. Let's run this. We're inside our debugger here. Let's print out what sequence repre is. Sequence representations, the 0th index of that, and that's going to be a tensor, and we want to know the shape of that. Okay, so there you go. This is our encoded. This is a tensor of size 1280. So ultimately what we want is we want sequence representation of 0. With index of that, and we want to put that on the CPU. I think it should already be on the CPU because we're doing inference on the CPU, and then I guess convert it to NumPy, and there you go. So I think that work. Let's put the columns here, or the comments on this example here. Okay. So we're copy pasting the comments on the ESM. Okay. Interrupt, then let's escape, save, here the output, and code the sequence column in the train data frame into a new column that we're going to call SM2. And let's go ahead and call this ESM2, or like the name of the actual head on that. This might take a second because it's doing every single sequence one at a time. If we actually wanted to do this quicker, we would do it in batches. Watch dash n is 1 in the SMI. So this is just we're looking at the GPU. Seems like we kind of are using the GPU. No, we never specified to use. And why don't we modify this? Well, so that, but eventually you want to use a different model in Alphabet, because I think really we want to use the biggest one that they have, which is, I think, this one, ESM2, 15 bill. But this one's going to take significantly longer to evaluate, so I think for the purposes of streaming on the internet, one is better. Bet ESM and that's the big boy model. Okay. See if ChatGPT is alive. Reset the thread. Can you write Python code that normalizes the EH and TM columns in a pandas data frame called train EF filter. Okay, so ChatGPT back. It's alive. Okay, I mean, I think there's an actual normalize function in pandas. I don't think you have to find the mean and the standard deviation and then normalize it that way. That seems kind of weird. Can you use the mean and standard deviations you above to also normalize the same columns in a data frame called test df? Let's see if we can use that as well. Look at it. It's clever. You can use the mean and standard deviation values calculated from the train DF filter to normalize the pH and TM columns in the test data frame as follows. So I'm going to copy paste the code that we had above. Perfect. Go ahead and paste that. I think we can do that. Code in between. Normalize the pH TM column, and we're going to also do this one. Okay. So I wonder if, can you write Python code for me that uses flip to encode a column of strings in my pandas data frame? So clip is a contrastive language image pre-training. And I basically, it's a good embedding space for strings, I think. So for the column here that's like data source or whatever, it's basically a string. And I want to encode it. Is there a way to encode strings with a large pre-trained model that doesn't use the OpenAI API? Such as BERT or GPT-2? can you write Python code to encode a string column in a data frame using BERT? All right, perfect. This is a Hugging Face Transformers library. Okay. This is still running, guys. It might not even finish by the time this stream is over, so go ahead and keep going over here. Let's see if we actually package Python. Okay, we do have it. Nice. Transformers, import BERT tokenizer, BERT model, and we want to code data source column. Why don't we do this? Like move. And I want to encode the filter. I also want to okay, so we normalize the pH column, we normalize the temperature maximum, which is the target that we're trying to get with our regression model. We also encoded the data source with a BERT model from the Hugging Face library, and then we are encoding the sequences with the ESM2 650 million protein model that face SDF, and I want train F. Okay. Now let's go back here and say, can you write Python code for me to train a regression model using features, or train a regression model on rows in a pandas data frame? The input to the model will be the columns ESM blah blah blah, then data source pH, the target for the regression model will be the column EM in a pandas data frame called train df filtered. Okay. The columns ESM and data source are vectors and need be converted into a proper data type such that they can be used. Let's see, that's a pretty complicated set of instructions that I just gave it there, so let's see if it actually gets it. Okay, here we go. Yes, here's an example of how you could train a regression model on rows in a pandas data frame called traindf filtered. Okay, it's going to use sklearn. Impute, pipeline, simple imputer, scalar, one-hot encoder, linear regression, numeric features, numeric transformer. Okay. It's not a categorical feature. We run all of these. Let's run everything above. Start. Save. Run everything. Test does not have a encoding the description column. Downloading the BERT model, which we'll use to encode the list object has no attribute size. Maybe data source is list. What is a filter? String, right? It's like a string. Maybe it's this BERT tokenizer. Let's see what Copilot gives. BERT base uncaged. Okay. Model. So VS Code is giving you that same one. Let's say load the data source column and encode. Tokenize and then encode. The tokenizer dot code, and then you feed it into the model. Tokenized. Let's see if this at least works. Get rid of this part here and rerun this. So while we're doing that, we're going to rerun. So let's going back and forth here. So sorry for, but let's look. Have this tokenized head. Open up a new ChatGPT. I am using a BERT tokenizer. Can you add, or can you modify the code below such that the tokens are fed into the BERT model? Tokens into BERT model. All right, let's see what happens here. So we kind of copied a bunch of code here that and we want ChatGPT to give us this next line here. And this next line, to feed the tokens into the BERT model, you can use the model object and its forward method. You will need to provide the tokens as input to this method, as well as the attention mask, which tells the model which tokens are real and which are padding. Here's an example of how you can modify the code to feed the tokens into the BERT model. Okay, it's copying our code, so it's got the tokenizer in the model. It's tokenizing the data frame, tokenized. This is correct. It's tokenizer. Okay, correct. And it's creating attention mask. So, okay, it's now taking the data source tokenized and applying int token ID greater than 0 for token ID in X. And then it's feeding the tokens and attention mask into the BERT model. Can you modify the code above such that the encodings are put into a new column called data source encoded in data frame? Yes, you can modify the code to create a new column in the data frame that contains the BERT encodings for the tokens in the data source column. So slow. It's a little slow, but this is still pretty amazing. And it's, you know, if I was starting again as a programmer, completely different skill set, you know, because now your ability to kind of interact with these AIs is really how you get the best results in the fastest possible time. I don't know if this will necessarily work, but we can see. Let's try something crazy here. I'm getting this error when I run the code above. Do you know what is wrong? You can simply rename the input IDs. Okay, I don't know if this is the actual solution to this bug. This seems kind of weird. And I don't know how it's going to change this at all. input IDs. Let's actually go Hugging Face BERT model. Going on the docs here. To actually see what's going on. BERT config, blah, 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 BERT tokenize, blah, blah, blah. Input IDs, token IDs. BERT tokenizer. TF BERT tokenizer. We want like example code here. Okay, here we go. Smaller. Return tensors PT. Maybe this is what we need. And then star import. Okay, so first of all, it's not tokenizer.encode, it's just tokenizer of X with return tensor. And then you want to do the model of star that, whatever that return. And then you want last in states. Coded BERT. Okay. Base model output pooling with and cross attentions does not have a last hidden state, so it's last hidden. My GPU away. That utilization on little hot tip whenever you're doing deep learning. You know you're doing the right thing when you hear your GPU start whirring in the background. Okay, so you just joined here. What we're doing is we're taking the data source from this Novo enzyme problem on Kaggle. This is what the data source looks like. It's like weird links and all text data. So what we wanted to do is convert these into a encoded, a latent vector, and we are using the BERT model from the Transformers Hugging Face library. We tried to get ChatGPT to do this for us, but it gave us code that didn't work, and then we tried to kind of fix that code, and it also didn't work. Then we tried to do it with GitHub Copilot, and it didn't work quite well either. So we went ahead and actually went into the Hugging Face Transformers documentation and API, and then inside of here they actually have an example, and then we just used that example. And I think this is going to actually give us the answer that we want. So perhaps cheating a little bit, you know, if we were trying to do this entirely with ChatGPT, we had to kind of resort to actually going to the answers. But it's looking pretty good. So unfortunately, like, it looks like this is taking forever because of course it's going through every single row in the training data. So I think what we're going to do, just so we don't sit here all day, is let's go ahead and get the training data and just reduce it. So let's say, can you write Python code to reduce the number of rows in a pandas data frame 100. Go ahead and put that here. We're going to put that up here so that we can actually run this notebook in a reasonable amount of time. Put a little. Remove this line before submitting for final training. Here are the outputs of all the cells. We're going to start this notebook, save, and then we're going to run all again. Okay. So now we're doing it only on a very small amount of the training data, so it should run significantly faster here. This is the code that we had beforehand. And want can you convert this column in the data or the column in this data frame into a NumPy list from PyTorch tensor. And this is still going, this code sequence. Maybe we just need to add the .cp.numpy. This part here. Go ahead and see. It's still running, so actually let's interrupt. And whenever we were filtering top. Let's filter to 10 rows. So this is impossibly small training data set, but we're just seeing how this works. And let's run all the ones up until here. Can't call numpy on tensor that requires grad. Okay, I think we need model.eval. With torch no grad. So we don't want a gradient, right? Run this. Torch is not defined. Right. Now let's see what. Okay, we have a nice little vector. And df filtered dot 10 numbers. Fine. Run. This is the code that encodes the sequence in a latent vector. To make sure that our expression is convert. Okay. Let's so we normalized t. Let's say n to perform. Can you add to the code below such that use reg model to predict the PM column of the test df frame, making sure unnormalize the predicted prediction. And we're going to give it the normalize the TM column. Just going to copy paste and repeat our code from above. Something's going on here, guys. Something's going on with this ESM pre-trained model. It's really not taking way too long. Even just 10. Station 4. Okay. Can you modify the code above? Put the predictions in a new column of the test df data frame. Also do another line here that can you write Python code for me that will clear out any stale data in my GPU? I want to run this code in a notebook. I think what might be happening is the GPU is already filled with BERT, so interrupt, clear outputs of all the cells. BERT. Back here. Put this BERT. There's BERT coded. Gotta print. Verted sequence coded this sequence coded interrupt. Okay, so now 5. 6, 7, 8, 9, 10. Oh, because it's also doing the test data frame. That's why it's taking forever. Okay, let's interrupt. Let's clear the output of all the cells, restart, and let's go ahead and test data well. Test name. Test df dot head and 5. Okay. Start, save, run all. Actually, we need to put the test L, test df. No module named as head and flat. Pip install output of all cells. Start. Run. Successfully encoded the data source with BERT. It successfully is encoding the sequence with ESM2. And done. We'll see our sklearn code. Okay, now let's run learn model. No module named sklearn. Oh, I think it's pip and what is that? Pip s. Port. Okay. Might have just crap. That's there. that. Setting an array element with a this. Can you modify the code below? The issue I have is that the columns blah 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 are lists of integer or lists of floats. Says see what ph is I think it's nan because we're only doing like maybe 5 examples, so the normalization just ends up dividing by can't normalize just a data set that only has one piece. Dot values x write code to flatten list or flatten and unnest array inside the column of this pandas data frame. Come on, ChatGPT. Get going. Okay, so we're trying to take these columns are the sequence and the BERT encoded data source, like these like lists of lists. So we want to just convert them into, try again. We want to convert them into something that's like more flat, that you can fit into this sklearn model. Not really what I want. sklearn. Can I use multiple feature columns? Or can I feed a column in a data frame into this linear regression model that is a list, or that is an array? Let's see if maybe what was wrong here is that we were using a pH. pH is man. Like maybe if we get rid of the pH column, fine. So I think can you write Python code for me that converts a column with an array into columns that each contain a single number? A data frame. Yes, here's an example of how you could convert a column in a pandas data frame that contains an array into columns that each contain a single number. Oh, look at that. I didn't tell it, and it still has the sequence, and it still has the name of the column. I don't know the length of the array. Can you modify the code above so that it finds the length of the array rather than just assuming 100 element array? So I wanted to replace this 100 here with the of the ESM tree. Output columns. Output. Okay, that's what we got here. 1280, which is the dimensionality of our ESM2. And now let's do the same thing, but for the data source. Copy that. The same thing for the data source and convert. Convert output calls. Copy. I think the convert latent factor. NumPy dot pattern. Flat array now. Run this with convert. Complaining because it's not. This is not vectorized. It's like each one at a time. And then there we go. Okay. So there's actually 17,000 columns for the encoded BERT. A little bit intense. But, you know, we're getting there. A lot of things we've done wrong here. BERT output calls and then output calls. Those are going to be our regression here. BERT output calls plus ESM2 output plus, let's not do P. Now. All right, we fit our linear regression model. Test F. F. Or do F. Block here. We'll put that in part here. Repeat flattening. Test DF. It's not test DF. sorry, test df. F. Correct. That. We're like over 2 hours here. Columns must be the same length as problem here is you're gonna. What if we just do this? Getting desperate here, and we're just removing more and more things. Same thing. Out. Yeah. SDF and the columns we ID. Actually, that is what the sample sub. Can you write Python code to convert this data frame CSV file called sub.csv? Okay, while that goes ahead, the problem we had here is that BERT, the output of the BERT is a different dimensionality. So this code that we have here, numpy flatten last hidden model is. So we need to go back to Hugging Face transformers. Let's do logits. Okay. Base model output with pooling and cross attention does not have logits. BERT model. Maybe BERT for pre-training is what we want. Lax BERT for token classification. Maybe that's what we want. Try this one. Let's try BERT. Click here. BERT for pre-training output has no models logits. We want five numbers. It's 5-dimensional, but then within each one, how many are there? This. Run our code down here. All right, that seems. Let's go back to ChatGPT. Write the data frame to output file. We want Output file, we want submission.cs. I'm here. Interrupt. On the end. I think we're going to call it a day once we. So this is our submission. This is the. Obviously fairly shite because we only ran it on, or trained it on 5 rows of the training data. But we're almost getting to the 2.5 hour mark here. So I think that call it there. Interesting results. You know, we did end up using ChatGPT and VS Code. There was a lot of stuff that we used it for. It did very well in the creating pandas code for us. It did significantly more poorly when we actually wanted it to create these kind of encoding stuff. So when we wanted to convert the amino acid sequence into a latent vector through the ESM2 model, it wasn't able to do that. And when we wanted to convert the data source, which is a string, into a latent vector that comes out of BERT, it also wasn't capable of doing that. And we had to go ahead and kind of piecemeal those things from the actual official documentation of BERT and then the official documentation of the ESM. So it's not quite there, especially with stuff that is, like, I think it's good with things like pandas, because pandas is a library that's been around for a long time. So there's a lot of examples on the internet and probably people asking for questions about pandas, and the answers are a lot more consistent. But for GitHub repos that are relatively recent, or things like BERT, where maybe there's a lot of different ways a lot of different BERT models, a lot of different ways, a lot of different functions and ways of loading and using the different outputs of the BERT model. So ChatGPT will get confused, right? Because there's a lot of different things to do there. So yeah, overall, interesting stuff, but not 100% there yet. You still need a little bit of kind of human intuition and elbow grease to kind of get it to do what you want. I'm going to go ahead and put this here. Commit enzyme stability. Add this. We're going to commit. And yeah, that was enzyme stability prediction on Kaggle with ChatGPT. Been great. This has been a long one, and thanks for listening, guys.